All right, I'm going to share my screen. All right, so welcome. This is a brief overview of what we're going to be covering today. So I'm going to briefly introduce the Allen Institute and then hand it over to Luke to introduce the Cell Types Database Research Program. And then Nathan will speak about the Patch-seq data set and Stephanie about the Synaptic Physiology data set. And we will have time at the end for Q&A. So again, put those in the Q&A button at any time. So a little bit about participating here in Zoom. Please remember to put those questions for the panelists in Q&A on Zoom or ask them in the YouTube chat. Please don't put your questions in the Zoom chat or else they might get lost. But use that chat to talk to each other about your research interests, introduce yourself, share observations and other comments. And please make sure you're sending those chat messages to all panelists and attendees so others can see your message. And we have live captions available in Zoom. And for YouTube viewers, those captions will be available on the recording. Our main presenters today are going to be Nathan Gowans and Stephanie Seaman. Both of them were involved in the development of the resources they're going to be sharing with you today. We're also going to hear from Luke Esposito, the Senior Director of Operations for the Allen Institute for Brain Science, on the cell types program that encompasses these data sets and more. The Allen Institute is a biological sciences nonprofit located in Seattle, Washington, with focused research areas in neuroscience, cell biology, and immunology. We also support cutting-edge research across the world with the Paul G. Allen Frontiers Group. Across our institutes, our scientific process is focused on tackling complex, broad, and hard problems in the fundamental biological sciences. And that approach is guided by these three core principles: big, team, and open science, which I'll return to in a moment. And our work generates data, knowledge, and tools like the ones we're going to talk about today, all of which we share publicly. To date, we've collected about 14 petabytes of data, which is about 2.8 million DVDs worth. So the most relevant of those three core principles today is open science, which is we share all of our data and tools for you to use in your work, and we want you to use them. So please check out some of the other webinars in the series that we've already run and that we will be running at this symposium in a few weeks to learn about how to use some of our other resources, as well as the two resources that we're going to be featuring today. A final reminder, please use the chat to talk to each other and introduce yourself, but use that Q&A panel to ask questions at any time. And then if you're interested in joining the Allen Institute team, we will drop a link to our careers page in the chat if you are so interested in these open resources that you would like to be developing them yourself. So I'm now going to hand it over to Luke to introduce the Cell Types program.
Okay, thanks, Caitlin. Welcome, I'm Luke Esposito. I'm going to introduce the Allen Cell Types database to provide context for the tutorial on the Patch-seq and Synaptic Physiology resources. So at the beginning of these presentations, we like to connect with our diverse audience. And we know that the researchers in the audience include current or new users of our Cell Types database at brain-map.org. Many of you are basic science users, but some of you are also focused on disease, translational work, or maybe even clinical research. And we know that computational biologists comprise a large portion of our users. And so we aim to provide useful information to all of you. So my brief introduction is broken up into two parts. First, what is a cell type and why are they important? And then second, I will introduce these techniques of Patch-seq and Synaptic Physiology that we use to identify cell types and probe their connectivity. So first of all, what is a cell type? So a cell type is a population-based classification which is dependent upon some discontinuous variation within the population. Cells can be grouped based on their similar characteristics. So cells that are in a group are more like others within the group than they are outside the group. Neurons can be grouped based on their similar morphology or shape, and this is work that goes back more than 100 years to the time of Ramon y Cajal, who drew these beautiful pictures of neurons in the cortex, cerebellum, and retina. Neurons can also be, or cells can also be grouped based on their similar task or function, although in the complex tissue of the brain, this is often not immediately clear. Cells can also be grouped based on shared features that are obtained by one or more measurable criteria, and these criteria are ideally quantitative and linked to function, and you'll hear examples of this in the Patch-seq portion of today's tutorial. Cell types are hierarchical, with a class at the top, followed by a subclass, and ultimately a type, and in some cases, a subtype based on the split of a type with new data. We think cell types should be treated as a testable hypothesis subject to change upon new discovery. So historically, the definition of cell types has been relatively coarse. It's been based on immunohistochemistry with relatively small numbers of available antibodies for those studies. But more recently, we've had several technical advances that enable a much finer characterization. For example, single cell and single nucleus techniques, next-gen DNA sequencing. So indeed, we're in the genomics era where we can dive deep into the gene expression patterns of cells, the transcriptome. There are engineering advances that enable us to use single cell techniques in a more high throughput fashion. And you'll hear examples of that in the multi-patch synaptic physiology portion of today's tutorial. We also benefit from transgenic technologies and tools that enable us to access and assess a subset of the many different types of cells that are present in the brain. We also benefit from high resolution imaging and three-dimensional reconstructions that enable beautiful pictures like the ones you see here on the right. And then we have increased computational power, statistical methods, and dimensionality reduction algorithms that allow us to process these very large data sets and to convert data to knowledge. So why do we care about cell types? Well, we think it's obvious. To understand the whole, we must understand the parts. While that's probably self-evident for your cell phone, we think it's also true for the complex tissue of the brain. In addition, we know that in neurodegenerative disease, specific populations of cells are lost very early in the disease. And so we think if we can identify what these cell types are, we can intervene early in the disease when intervention is more likely to have a positive benefit. But our first goal is to identify and characterize cell types in the healthy brain, which is really the main focus of today's tutorial. So what do we mean when we say we can organize cell types into hierarchical clusters? So imagine you have a population of cells shown here on the left. Each dot is an individual cell. You then apply a set of measurable criteria and use a computer algorithm to group cells based on their similarities or differences as indicated by this color coding. The software then can generate this dendrogram or tree-like hierarchy of clusters, which is shown here on the right. So cells that form a type these clusters are really what we define as a type. So cells that are members of a cluster are very similar to one another. They are similar to, but slightly different than cells in neighboring clusters and significantly different from cells that are in more distant clusters in the dendrogram. So in the last minute, I'll just introduce these two methods of Patch-seq and synaptic physiology that we hear about in much more detail from Nathan and Stephanie. And we use these to identify cell types and then understand how these cell types may connect. So for Patch-seq, we aim to go from single cells to multi-dimensional cell types. We collect data around 3 different cellular properties: gene expression, which is the transcriptome, the shape of the cells or morphology, and then of course the firing patterns, which is electrophysiology. We then take all of this data and use joint cluster analysis, which considers all 3 modalities, to generate these multi-dimensional taxonomies or MET types that come from the Patch-seq data. It's one thing to understand the parts, but we also have to know how those parts connect. So to do that, we can probe connectivity using this technique, multi-patch electrophysiology. And here we use genetic tools to access a subset of the cells that are in the brain and then assess the probability of connections. And if there is a connection, the connection characteristics. And you'll hear about that in the second part of the tutorial. So with that, I will stop sharing and hand it over to Nathan.
Great. Thank you, Luke. Let me just pull up my screen. Okay, so that was a great introduction by Luke about why we're interested in these cell types and the different methods that we're using to try to understand and characterize them. So as Luke mentioned, the advent of high throughput single cell transcriptomics has really expanded what we can do in terms of defining cell types. So this is a technique where you dissociate cells from a tissue, you figure out what genes are currently being expressed in them, and then you have this really high dimensional readout of kind of the state of the cell and its characteristics. You can use that high dimensional information, which is this is also a high throughput process. So you can get many, many cells and do things like, use one of these nonlinear, embedding techniques to just kind of display the basic transcriptomic structure that defines different types of cells. And so you can see like large classes like GABAergic inhibitory neurons forming their own continents and glutamatergic excitatory neurons forming other continents. And then you can look at the more granular structure and identify different types, cell types, transcriptomic cell types that have particular expression patterns that differentiate them from each other. You can also take that and form a hierarchy, like Luke mentioned. And when you do that based on this transcriptomic information, you see that it recapitulates a lot of what we kind of already understand about cell type organization. So, one of the major splits in the trees is between these GABAergic inhibitory cells and glutamatergic excitatory cells. And then from there are, further subdivisions that correspond to major markers that have been used to identify different classes of cells or different laminar locations. But this view provides us even more granular, kind of even more numbers of cell types than we would have just by using what historically has been used to distinguish cells from each other. So that kind of brings up the question, how do these transcriptomic types correspond to phenotypes that we already know about? Which of these transcriptomic types corresponds to, a basket cell or a Martinati cell, and how different are these different transcriptomic types from each other? So in some cases, you can make that connection pretty clearly. Like, for example, this parvalbumin Viper2 cell type expresses a set of markers that are pretty unusual and unique and correspond to the well-known, also quite unusual cell type known as the chandelier cell. But in other cases, maybe among the different SST types, you don't know which ones are the Martinati cells, or you might have a cell whose molecular identity is somewhat ambiguous and you're not sure if it really has more neurogliaform properties or more of chandelier type properties. So in order to figure out how these different types correspond with each other, you really want to collect information about these different phenotypes from the same cell. And so that's where the Patch-seq technique comes into play. So the Patch-seq process was developed by a few different labs, and then we at the Allen Institute tried to make this a more kind of standardized and high throughput process to get a big data set about cells from a particular part of the brain. So it's based on regular kind of whole cell patch clamp electrophysiology to start with. So in that case, you have a slice of brain tissue. At the Allen Institute, we either take that from a mouse, usually a transgenic mouse, where a particular subset of cells have a fluorescent reporter, so we can target that. That's why the cell is in red here. But we also do the same kind of experiments from human neurosurgical tissue, so we can characterize cells from humans. And then you take a electrophysiology pipette, which has an electrode in it and is filled with dye, and you bring it through the tissue and patch onto the cell, and you form a high resistance seal against the cell, and then you rupture the membrane underneath that pipette so that you get electrical access to the inside of the cell, and also allow that cell to be filled with dye. So when you have this electrical access, you can go through an electrophysiological protocol, which we do the same thing in all the different cells, record their spikes, record the different properties, subthreshold properties. And then when you're finished with that recording, after you've characterized the electrophysiology, we apply negative pressure, so suction to the pipette, so that we can extract contents from the cell, including we try to get the nucleus and pull a nucleated patch, which you can see kind of being schematized here. And that leaves most of the cell intact, again, with the dye still there, but also gives us the contents, which that we can then take and do the same kind of transcriptomic analysis that we do with those dissociated cells that I mentioned a couple slides ago. And then we stain the remaining, the two, and image the cell so that we can reconstruct its morphology. So through this technique from the same cell, we've got their electrophysiology, the morphology, and the transcriptomic information. So the first thing we want to do is just see what we can do with that transcriptomic information. The approach that we've taken is to use this. So this is not as high throughput as like just dissociating tissue and all the cells you get, because you actually have to patch them. we wanted to use the larger data set from the dissociated cells as a benchmark, and then use that to define the types, and then map our Patch-seq data to those types. And so this is just showing these different types in different columns, and we're showing that the gene expression patterns that we see are very similar between the dissociated cells and the Patch-seq cells. And so the same types are expressing the same set of markers, which aren't necessarily being expressed in other types. And overall, the data quality looks quite good. We're quite happy with the mapping. There are a couple differences, which I won't get into. You can definitely see our publication, which came out last November for details. But overall, we think that we can pretty reliably map most of the cells to a particular transcriptomic type. So we don't want to just look at the gene expression information, although that's interesting. We want it, because we're doing Patch-seq, we have other information as well about the other properties. So for example, we can look at the different electrophysiological responses and sort it by transcriptomic type. And so you can see that there's a range of responses from this late spiking, firing, to fast spiking cells, to more irregular spiking cells. And those differences are, it's quite diverse across the population that we've looked at, which are GABAergic interneurons in mouse visual cortex, consistent within a particular type, which gives us some confidence that splitting transcriptomics is a reasonable thing to do. We can also look at the morphologies, and you can see kind of a similar story here. Again, there's quite a diversity in terms of the shapes of the cells, in particular their kind of axon innervation patterns across these different types. But there are also strong similarities between cells that come from the same transcriptomic type. So we want to use all of this information together to define legend kind of types based on 3 of these modalities together. So these morpho, electro, transcript types, or MET types. So I'm showing just an example of doing that with the LAMP5 subclass of cells. And so you can see here we've partly because of just the data we've defined 2 particulars. So one tends to be more superficial and has a little bit of a variety of morphologies, and the other tends to be deep, but both have kind of similar late spiking electrophysiological properties. And see where they fall on a transcriptomic 2-dimensional embedding, as well as an electrophysiology 2-dimensional embedding. And you can different things. So you can look at these cells and see how homogeneous or heterogeneous they are. So in this case, we find that there's actually kind of a continuum of properties, but those continuums line up with each other across these different modalities. So there are some cells that express a lot of NPY and other cells that express less NPY. And the cells that express a lot of NPY tend to have more of a round shape. So their width to height rate closer to 1. So they're more like this kind of cell. And cells that have low NPY tend to be wider than they are tall. And the ones that are more ball-like also have a delay to their first spike. And the ones that are want to have less of a delay. So what I'm going to do now is switch over and show you how to access this public data. I think you can do analyses like these or other, really interesting analyses with, and show you how we, from a particular example cell, then you could apply that to all the cells that you're interested in. All right, so I'm going to switch over to the web browser. All right, so right now the data is available publicly, but is kind of distributed over a couple different types. So we do not yet have an integrated web application to this data. We are working on that. We're really excited to get that to users, but it is not out yet. So right now, the way you have to do it is kind of going into figuring out where to get this from. And what I'm going to do is really kind of get into the nitty-gritty with that, how in Python, in a Jupyter notebook, you can access this data and analyze it. So if you go to map.org homepage, you can see that there's a link to Patch-seq data right at the start, so you can click on that. And then, like I mentioned, we have some data from mouse primary visual cortex. There's a kind of over 4,000 cells that are available there. We also have data from layer 2 and layer 3 human cortex, where it's primarily glutamatergic cells. We've also made specific protocol that we're using to collect these data available online, and you can find there's a bioRxiv preprint outlining that, as well as a GitHub page that has different tools available, so you can do a similar technique in your own lab. We're going to focus on the primary visual cortex. I'll click learn more just to see what different files are available. So, this is just an example visualization showing how we're corresponding, how we're defining the different types. And we have electrophysiology and morphology from the same kinds of types. This is showing kind of on a cell by cell basis. You have a particular type that the cell's been mapped to. It's electrophysiology, it's morphology, and it's MET type. So down here is where you can access the different data. There's a couple different files that are particularly important. So one of them is this table of metadata, which includes information about where that cell came from. So it's brain structure, it's the mouse and the transgenic line, as well as the cell assignments that we've given to it. So the transcriptomic type and the MET type. There's also a file that just gives more extensive download instructions. So I'll go through it a little bit quickly when I'm doing this example, but you need that more to get more detail about how to get data from these different archives. There's also one thing that's very useful for getting that data is this file manifest, which basically says particular cell, a particular identifier, and a particular file type, what is the URL for that file, and that's how you can download it. So I'll show examples of that in a second. And then these are just directories to the raw data at these different archives. So NEMO is hosting our transcriptomic information. The DANDY archive is hosting the electrophysiology, and the BIL brain image library is hosting the neuronal reconstructions. Okay, so now I'm going to switch over to the Jupyter Notebook. So the first thing we want to do, so we've just kind of imported, our regular NumPy and Pandas libraries for accessing these file types. And the first thing I'm going to do is I've already downloaded the metadata file here. And I'm just going to open it and look at it. And so you can see when you look at these columns, there's cell specimen ID, which is kind of the main identifier for a particular cell. So each of these rows is a different cell. And then the columns are different metadata about that cell. Hemisphere structure, brain structure. And then here you see the different transcriptomic type labels or MET type labels that have been assigned to these different cells. And notice that one of them doesn't have one. That's because we don't have a morphology for all of the cells in the data set. So only cells that have a morphology have an MET type assigned. Okay, so then the other file we want to grab right away is the file manifest. So again, this kind of follows the structure I just described. So where you have the cell specimen that the file belongs to, its URL, and then the particular file type that it is, as well as some additional information. So you can use that to grab the URLs to get the files that you're particularly interested in. Okay, so I've also already loaded and actually loaded the transcriptomic expression, which is basically we can just take a quick look. Each row in this file is a different gene, and each column is a different cell. It uses a transcriptomic cell identifier. We'll link up to the other identifiers using that metadata file. And so the first thing I'm going to do here is just create a two-dimensional projection of the data like the one that I showed in the slides to see the structure, again, of those different subclasses and types. So I'm going to use the UMAP procedure to generate that embedding. I've actually already run it because it takes a little bit of time. So I'm just going to then load my libraries for plotting, so matplotlib and Seaborn. And then I'm going to plot the embedding. So you can see that there's a couple different islands. Those basically correspond to the major subclasses. Because I've looked at this before, I know that like this is where the SST cells are. This is where the parvalbumin cells. But we can use the metadata to see where particular types fall in this plot. So for example, let's say we're interested in the type 5 PLCH2 doc5. We're going to use the metadata file to identify all the places where the T-type label matches the cell type that we're interested in. So you can see the results of that here. So we have 100 cells, that particular type. And then we're going to use that to identify the particular cells in the gene data and figure out which cells to highlight with a different color. And so again, we have our whole map here, and then the cells in orange are those cells that belong to that particular LAMP5 T-type. And you can see they are mostly confined to this particular region of the UMAP expression, UMAP plot. We can also do things like look at gene expression, how that varies across this, embedding this two-dimensional space. So, we were interested in NPY from that example I showed you. And so we can see how that varies across the space here, just by coloring by the NPY value from our gene data. And you can see that it does also vary across the space. So it seems like there are parts of this LAMP5 area that are very high in LAMP5 expression, others that are lower. But NPY is also expressed in a couple other places in the UMAP. Okay, so like I said, so this is all transcriptomic information. Again, we could do that with a bigger data set, from the dissociated cells, but we're interested in other modalities as well. So we want to find, for this example, cells that have both electrophysiology and morphology data. So we can, again, use the metadata to find that. So we know that all the cells have both transcriptomic and electrophysiology. We want to also pick out the cells that also have morphology. So again, we're going to find the cells that have our particular T-type, and then also cells where we have a full neuronal reconstruction. And we the kind of information, and we'll just first cell that we get, so we can see its specimen ID here, as our example cell that we're going to calculate its properties from its electrophysiology and morphology. Okay, so now that we have this specimen ID of a cell that we're interested in, we're going to try to look at its electrophysiology. So the first thing we need to do is use our file manifest to find the rows that have that belong to this cell and that are of the NWB file type. NWB stands for neurodata orders. It's a standard format for electrophysiology data. Okay, so we can just see what that URL is. And then to get that cell, we're actually using, so it's in the Dandy archive. Dandy provides a command line tool to download data. So we're going to just use that to get that NWB file using that URL. Apparently there's a newer version available. Okay, so we've downloaded that file. It's about 15 megabytes. All right, and so we can also, get the file path from that file manifest. And what we're going to do next is use one of the libraries provided by the Allen Institute called IPFX for Intrinsic Properties Feature Extractor to load that NWB file, extract a standard set of features based on the standard protocol that we use at the Allen Institute, and then start to look at those features. So I'm just going to run that here. So again, I'm loading in the file from our NWB file. I'm going to drop failed sweeps so we don't accidentally try to analyze those. And then I'm going to extract the basic features from that cell. So this will run, take a second. You can see there are different types of cells that are being, or different types of responses that are being analyzed. So responses to long squares, which are a second long current pulse, short squares, which are a half second long or half millisecond pulse, or no, sorry, 3 millisecond current pulse, ramps, which is a slow current injection. And that's done now. So this basically tries to analyze all the kinds of things that you might be initially interested in about a cell. So we've divided it into those different, so this is a big dictionary, kind of nested dictionary of all kinds of different properties. We start out by just dividing it by those different protocol types. We can look to see what's available within the long squares. Okay, we see things like the individual sweep information as well as the baseline voltage, things about the Rio-based sweep, input resistance, membrane time constant. And so let's say we're interested in the Rio-based sweep. Okay, we got a lot of specific information just about that sweep. And then maybe we're interested in that latency to first spike, which is what was plotted in that figure that I showed in the slide. And so in this case, that latency is about a second from the stimulus to that first spike. And we can check that. We can plot the particular sweep that we've been looking at here. So we can let that out of the set using the sweep number that was identified from the analysis. And then we could just plot it by plotting its time and voltage. And so you can see here it is. Here's where injection starts. Here's where it starts firing. And you can see again that firing is about halfway through this one current step. Okay, so then you could do this with all the different cells that you're interested in and get all of those latencies out of it. So now we'll turn and look at the morphology. So we'll do the same kind of thing. We'll figure out where the SWC is from our file manifest. Now we're looking for this transformed SWC file. There's our URL, and we're just going to download it. Great. Okay, and so now, we'll get our path again. And now we're going to use a different library from the Allen Institute called neuromorphology. to load that SWC file into a morphology object that we can interact with. And I'm getting these different compartment types out of that so that I can plot the different compartment types in different colors. So I'm just going to make a quick plot, or I need to load the morphology. Okay. And then I'm going to make a plot by first buying out the axon compartments, the dendrite compartments, and the soma compartments, get their X and Y coordinates, and then plot those in different colors. All right, you can see that here. So the soma's in this black dot, the dendrites are in this orange, and then you can see that is extending, wide, a little bit tall, but yeah, this has kind of a similar shape that we saw in some of the examples in the slides. Okay, and then we can use the neuron feature library to also extract its features. Sorry, I actually didn't mean to run that. I had it already run. A second. But this is the last thing we're going to do. Oops, sorry, that's what I just did. Okay, and as soon as that finishes. So again, this is doing the same kind of like we did with the electrophysiology. It is calculating a whole bunch of features, kind of the things that we're probably interested in. And these are the kinds of features that we use to define the different morphoelectric and transcriptomic subclasses. All right, it should just finish in a second. Sorry about that. Just did it automatically. And so then once it's done, we'll put the width of the axon, which is just kind of coded in this particular format. So there's kind of nested features, so we can look like all of the nodes of the axon, or just the tips of the axon, or just the branch points, and then we'll calculate that width height ratio and see what. Or maybe I won't wait till it goes. So from when I calculate for, I'll just pretend that oh, there we go. Yay. Okay. I just had to threaten it. So, so you can, the height, or sorry, the width is about 700 microns. The height is 270 microns. And then, so it's about close to 3 times wider than it is tall. So again, you can do this kind of analysis on all the cells that you're interested in and construct a like I showed in the slide, and see how these different properties, correspond with each other across this multimodal data set. All right, well, I'm going to finish up there. Please definitely ask questions that we'll try to get to, but at this point, I'm going to switch it over to Steph.
I'm muted. There we go. Hey, everybody. I am Stephanie, and I'm a part of the Synaptic Physiology group. And hopefully you can all see this screen now. We're interested in sort of taking all of this interesting information about cell types and looking at how they're connected to one another, and thus the circuit functions that they play in the brain. And so we're familiar by now with this idea of there being multiple different types of cells in the brain, and they can be described by their morphology or their electrophysiology or their genetic expressions. And so these identities are hugely important for their role in the brain, not only these intrinsic identities, but also how they are connected to one another and the specific connections that they make with other types of cells and the properties of those connections. And so that's our focus in the Synaptic Physiology group, is to understand this local microcircuit. And so how do we go about doing this? We use some similar techniques that Nathan described to you. In mice, we use these transgenic animals that allow us to label particular subclasses of cells. And so we can do this with looking at particular types of excitatory cells, such as those in layer 5 that express this TLX3 driver, or particular types of inhibitory interneurons, such as those that express somatostatin. And so unique to the Synaptic Physiology project, we created mice that allow us to label 2 different subclasses within the same animal that allow us to target these specific interactions that would exist between these 2 subclasses. And to do that, we similarly use patch clamp electrophysiology, and we can record from multiple cells at once, hence the name multi-patch clamp electrophysiology. And so we can inject current into one cell and cause it to spike and record other cells at the same time and look for evidence of postsynaptic responses that would indicate a connection between these two cells. And so when we are in this sort of regime, recording from two cells, we'd be able to explore the two connections that exist between them. But we wanted to scale this up and see multiple connections at once. And so we will record from up to 8 cells at a time. And this allows us to probe the possible 56 connections that exist among this group of 8 cells. When we do find a connection, we can quantify the different properties of the synapse. For instance, we can see if the connection is inhibitory or generates an IPSP, or whether it's excitatory. We can measure the amplitude of these responses and get an idea of the strength of this synapse. We can measure the time course or the kinetics of the rising phase, or the rise time, or the decay phase, the decay tau, to get an idea of the kinetics of these responses. And we can look at how these responses evolve over time. And so during an ongoing spike train, if a synapse shows a weak response at the beginning of the train, but this response grows over the course of the train, then this would indicate a facilitating synapse. Conversely, if the response starts out strong and grows weaker over time, then this would be indicative of a depressing synapse. So these sorts of metrics, the strength, kinetics, and short-term plasticity of both inhibitory and excitatory synapses, are the types of things that we measured when we found connections between different types of cells. And so we did this for individual synapses, and then our goal was to accumulate these across different types of synapses and to form a more global picture of what the circuit looks like. And so we can imagine that if we view this as sort of a matrix of interactions, we can split out the different layers of cortex, layer 2, 3 down to layer 6. And then within each layer, we were focused at what is the subclass level, which primarily means that we had about 1, possibly 2 excitatory subclasses within each layer. And then looked at the 3 main inhibitory subtype or subclasses in each layer, mainly parvalbumin, somatostatin, and VIP. So we have these different subclasses for each of the layers, and we can organize it such that we look at presynaptic cell subclasses on the rows. For example, we could pull out this layer 5 SST subclass, and then look at the postsynaptic subclasses along the columns. In this example, pulling out the TLX3 pyramidal type, which labels a layer 5 IT or intratelencephalic subclass. And so then the intersection of these 2 row and column are the unique element that describes the connectivity and synaptic properties of a layer 5 SST cell connecting to a layer 5 IT cell. And so with this matrix framework, we can layer all sorts of data on top of it in the form of heat maps. And here I've shown a couple of examples of that. So we can look at just connectivity, how likely cells our particular subclasses are to connect to one another. And so here we can see that the warmer colors, the yellows and oranges, show high connection probability, whereas cooler colors are lower connection probability. You may notice that we have a bit more data along the diagonal here, and that was because we mostly focused on intra-layer connectivity, so looking at the circuit within layer 2, 3, or within layer 4 or layer 5. And so that's where most of our data lies. You know, by accident, we sometimes got these off-diagonal inter-layer connectivity elements, and those are super interesting to look at as well, but we'll have less data there. There are other projects at the Institute that are looking a bit more at these sort of off-diagonal elements, and it's something we might explore in the future ourselves. We can look also at the strength of these synapses, where red elements here denote excitatory synapses and blue inhibitory synapses. And similarly for short-term plasticity, red elements would be those that are facilitating and blue, those that are depressing. And so we provide all of these data for you to interact with on your own and explore. And there are a few ways that you can go about doing that. We have a main web page that I'll walk you through, as well as some Jupyter notebooks to play with. And we do have some downloadable interactive tools if coding is not so much your jam. And you can download these and sort of view the data as I showed you in these matrices and click around and explore the data that way. So I will share now the website. Okay, so here we're back to the main brain map website, and the best way to get to the synaptic physiology page is to go to this explore and then connectivity matrices. And so on this page, you'll see that there's actually a couple of data sets and projects highlighted. The Synaptic Physiology group is one of a few that are interested in looking at connectivity across the brain. And so there's also the mesoscale connectivity, which looks at connectivity across brain regions, as well as the ultrastructural or EM connectomics, looking at that fine scale connectivity as well. And I think they are giving a tutorial in the coming weeks, so you can learn about that. And then down here at the bottom is our synaptic physiology data set, which we can click on to enter our main page. So here we have a lot of information for you to explore, and I'll let you all do that on your own. You can get a more sense of what exactly is in the data set at a higher resolution, and down here learn a bit more on the methods that we used to both get the data, the experimental methods, and how we analyzed it. If you want to keep in touch with us, you can always reach us at the community forum or sign up for our mailing list. For now, we'll pop over to this how to access the data page and get you going on how to play with this data. So as I mentioned, we also have these Jupyter notebooks that you can use. We've put them into this MyBinder, which is a nice way to organize these notebooks and allow you to get going right off the bat. The MyBinder will download the data that you need, as well as load up all of the Pythonic environments and repositories that you need. So all you have to do is click this button and get going, and we'll do just that. So this does take a minute to load up, so I've done that for us already. And it will bring us to this page here, which you can see we've got a handful of notebooks pre-made for you to start playing with. And we'll look at this connectivity notebook to start. Oops, that's not good. Okay. So here we're looking again at that connection probability. So the likelihood that cells of different subclasses are connected to one another in that matrix that I showed you. And so again, we're just gonna first import a bunch of libraries. We have all of our data stored in a database that's downloaded here for you. And we've also made a few ease of use functions and classes that get you going. And so here in this first cell, we'll load up the Synaptic Physiology database, and this is the particular version that will get you the data we need. These are SQL databases, and so all standard SQL queries and functions will apply. We've made a few that are specific to our data set. One of these is this pair query. So our data is highly centered around this idea of a pair object, which is a presynaptic cell and a postsynaptic cell that were recorded from at the same time, and thus all of the information that goes along with that pair. So here, we're just gonna query all of the pairs that were from mouse data or mouse experiments. And so we'll get a list of all of those pairs. And if we pulled out the last pair in that list, we can look at a bunch of information about it. And so here's its unique identifier, the experiment ID, plus the presynaptic cell number and the postsynaptic cell number. In this case, the presynaptic subclass was a parvalbumin cell, And the postsynaptic was this NR5A1 Cre type, which is a layer 4 excitatory type. And there was not a synapse detected between them. If we pulled out another pair, in this case, I knew it was pair number 504, we can see that this was a pair in which it was a SIM1, a layer 5 ET type, SIM1 to SIM1 pair. And in this case, there was a connection between them. And so then we can explore more things about it. And so you can see in these lines up here that the pair object has many different attributes. It's pre-cell and it's post-cell, but also has this synapse attribute if a synapse was detected. And then that comes along with many metrics describing that synapse, such as the amplitude or the latency. And so we can see that this connection was quite strong, 1.5 millivolts almost, and the latency to the onset of the EPSP was 1.3 milliseconds. There are many other attributes of the pair and the synapse that you can explore by looking at our documentation, which is linked off of our website. So once we have all of these pairs, we want to group them in reasonable ways. And so we do this by using this cell class object that we created. And so this is a nice flexible way to define cells however you choose. All you need to do is specify the types of criteria that you're interested in. So in our example here, we were interested in looking at the layer and the Cre type, which we use as a proxy for subclass. And so here then we can say that we want to define layer 2-3 PV cell class as expressing parvalbumin and being in layer 2-3. So we repeated that a bunch of times with different types of subclasses. As you can see for layer 2-3, you could also use things like dendritic morphology to describe these cells. And so you can construct these cell classes at any resolution you choose and with any combination of features. So once you've defined your cell class criteria, here we can create the cell class object. And once we do that, we can then group all of the cells into their particular cell class, explore the pairs that exist between those cell groups, and then measure the connectivity between them. And so here's where we get a dictionary of results describing the connectivity between all of the possible combinations of these cell classes. And so in that dictionary, we could pull out one element, which in this example is the layer 2-3 SST to layer 2-3 VIP connection, and which is an interesting one for disinhibitory circuits. And we can print out a bunch of things, such as there were 4 synapses found out of the 27 that were probed. That gives us a connection probability of 14.8%. And we also calculate a confidence interval around this connection probability. So using all of this information for all of the different cell classes and their interactions eventually gets us to these matrices that I showed you in the slide. And so here again, you can see that layer 2-3 SST to VIP element where we saw 4 out of 27 synapses were found. And the colors indicating the likelihood of connection. And then the saturation of these elements is determined by that confidence interval. So elements in which the color is darker, it indicates that we had a lot of experiments there. We probed a lot of connections and our confidence in that connection probability is quite high. Whereas those that are more opaque, we have less data for, and so our confidence on that connection probability is a bit lower. So as I mentioned in the slides, this is showing just connection probability. You can do similar types of analyses with strength or short-term plasticity, and the other notebooks go into that. So I'll just scroll down a little bit and also highlight that we also recorded from human neurosurgical tissue. And so you can similarly access that data here by pulling out pairs that were part of human tissue projects. Again, we don't have transgenic humans, so we can identify cell classes here mostly by their dendritic morphology. So in this case, we could look at all of the spiny or excitatory cells in the different layers of human cortex and do a similar analysis looking at the connectivity among these to get a connectivity matrix for human excitatory cells. And so we can see here that we've got pretty high connectivity in the upper layers, layer 2 and layer 3, with fewer connections down in the deeper layers. One last thing I'd like to point out is, our data set goes well beyond just pure connectivity. And one element that's particularly interesting or good to keep in mind when thinking about connectivity is that it often is very highly related to the distance between cells. Many people have studied this before, and so we also allow you to do that. The pairs in our data set have distance, intersomatic distance between them. And so if you run through all of these different cells, at the end, you could look at how connection probability falls off or remains constant with intersomatic distance. And so here we just pulled out the 3 main inhibitory interneuron subclasses, and we can see that parvalbumin cells, while they have high connectivity when the cell bodies are close to one another, as you get further away, these connections pretty dramatically decrease. Whereas connections among SST cells or among VIP cells have a low connectivity overall and is pretty constant with distance. So that was the connectivity notebook. I'll let you all explore some of these other notebooks on your own. And again, if you have questions or want to get in touch with us, there are ways to do that back on our main website. And now I will throw it back over to Caitlin.
Yeah, thank you everyone for presenting today. So we've had a very good number of questions in our Q&A here on Zoom, and those of you who are watching on YouTube, we will take questions from you if you have any. And many of those questions we have already addressed by typing out answers. So if you have not gotten a chance to read through those, we will post all of those questions and answers on our community forum and link to that from the YouTube video description after today's talk. So you can catch up on all of those answers later, including several of those answers have links. So we have one question that's for you, Steph. What was the prevalence of autopsies, self-innervation in your data set?
Yes, so unfortunately we did not look at autopsies. They're a bit hard to find considering they'll be very close up against the presynaptic spike. And so we have not looked at that yet. All of our data is available for you to download. So if you would like to go dig into that yourself, I would highly encourage you to do so.
Great. We have another question for you. What patch solution do you use to record facilitation and depression simultaneously? Do you use cesium methane sulfonate? And I've already included the link to your protocol, so if you can just speak to that piece specifically.
Yeah, I would say go look at the protocol directly, but we do not use a cesium-based. All of our internal solutions are potassium gluconate-based.
Great. We have another question that just came in. Does the nuclear extraction take place soon after stimulation? I'm wondering whether immediate early gene activation is captured with this protocol.
Yeah, so yeah, basically right after we finish the stimulation protocol, we start extracting. The extraction is fairly slow, just partly to make sure we get the nucleated patch. But still is like, a couple minutes at most, I think. So probably some immediate early gene activation going on. We do think overall we see a little bit more immediate early gene activation in our Patch-seq data set compared with our dissociated cell data set, although there could be just activation by the slicing and stuff like that too in that data set as well. So it doesn't seem to affect the mapping. I don't think a lot of the mapping is terribly, those are not generally the marker genes used to define the types, but it is something interesting that you could investigate.
So this is a question that you had already answered, Nathan, but just for everyone to hear this answer, because I think it's a common question. how big are the slices usually to get a reliable 3D reconstruction? And also for Steph, how thick are the slices used in these recordings?
For Patch-seq, it's a 300 thick slice. And so we know that parts of the morphology will be cut off. You can very clearly see that when you do the reconstruction. That's kind of inevitable for slice physiology. We do like, so for the ones that are kind of extend over longer periods of time, longer distances, like Martinati cells, or when we do excitatory cells, like their apical dendrites, we try to make sure that those are intact and in plane. Just a variety of cutting angles from the brain to try to make sure that that maximizes kind of the in planeness of the cells. So we get as much of that as possible, but we will fail morphology if it's too truncated. So.
Yeah, we use the same slice thickness, 350 microns, and employ all the same techniques that Nathan described to make sure that we're getting full axons, which are important for finding connections. And we try to record deep in the tissue. And then we will, in our data set is also information about the depth of the cells that were recorded from, along with how long the axons and dendrites were before they were truncated, if they were truncated. And so we can look at all of that information and how it might affect the connection probability of different types of cells and correct for that if needed.
So this question I think is mainly for Nathan. How many nuclei can you extract per slice? And to modify that question for Steph, how often do you actually get all 8 cells that you can record from?
Yeah, so you know, it depends a little bit. It depends more on kind of like the slice itself and like what regions we're targeting. Like so. For humans, like we try to get as much data as possible. So there can be like, I think even they've recorded from like up to a dozen different cells. Mostly you just want to space it far enough apart that their morphologies are not overlapping and so that they'd be distinguishable. And if you kind of, you know, the regions are big enough that you are getting cells from the region you're trying to target. In the mouse, it's a little bit more constrained just because like the region you might be targeting is a little bit smaller of a part of the slice. But we're also interested in going beyond visual cortex for using Patch-seq. So in that case, we'll probably get more cells per slice too from a variety of regions. Yeah, and one thing to just clarify. So what we're doing is we're pulling a nucleated patch. So we're not actually extracting just the nucleus. There's definitely cytoplasm that is coming along with it. It's basically like you get kind of a micro, you know, around your nucleus that you're pulling away. So it is not like it's not like we're, you know, something where we're trying to actually get rid of all the cytoplasm and just get the nucleus. It just helps us to get the nucleus and probably all of its, you know, transcriptional machinery that's kind of glommed onto the nucleus as well. So just to be clear.
Yeah, and so to speak to the number of cells that we record from at once, we pretty routinely get at least 4. That is probably our median. But we have gotten, I would say, maybe 50, what we call octo patches, or all 8 across both mouse and human. So it happens, it's a rare, but we're getting better at it.
Well, we're just about out of time for today. Before we go, I just want to mention that you can find all of the data and the documentation at brainmap.org. Please do explore that documentation. And if you have questions, we have our community forum, which you can find by clicking on updates and support in the top right corner of brain-map.org. And it's in that menu, or you can just go directly to community.brain-map.org. We'll make a post with all of the Q&A from today, including there's one question that we didn't get to. We will answer that. So you can find all of that, including the links after the fact. I hope you'll join us for our upcoming symposium and more tutorials. Please reach out if you have any more questions, and we'll post that forum post in the YouTube video description as well, so you can find it from there. And good luck using this data set. Happy analyzing. Thank you for coming today, and we will see you at future events.